Okay, hello everyone. Thank you for joining us to today's El Zoominario talk, which hosts today Daniel Ojeda-Juarez, who is with the University of California, San Diego now. Just so you know a little bit about him, Daniel was born in Oaxaca, Mexico, and then with the Rotary Club, he came to the US to finish high school in Michigan. Then he got his bachelor's degree in biology, and after that, he moved to San Diego to do his PhD in biomedical sciences at the Sanford Burnham Prebys Medical Discovery Institute, where he worked on HIV-induced brain surgery. And right now, Daniel is a postdoctoral fellow at UC San Diego, where he studies neurodegenerative diseases. And so thank you so much for joining us today, Daniel. It's a pleasure to have you, and please feel free to take it away.
Thank you, Daniel, and thank you, Claudia, for inviting me to present today. I'm really happy to be here and presenting. Thanks for the fantastic introduction. Today, I'll be talking about understanding neurodegenerative diseases, from Alzheimer's disease to mad cow disease. So just thank you for that introduction, but I also want to talk a little bit about myself. So I'm originally from Oaxaca, Mexico, all the way down here. It's a state famous for its pyramids, its culture, and its delicious food. I am really happy. I'm proud to be born in Oaxaca, and I had a lot of opportunities growing up locally with the Rotary International. They gave me a scholarship to go to Michigan to finish high school. After that, I was able to get another scholarship, and with my parents' help, to go to Fresno State to do a bachelor's in biology, in where I did cancer research. And then following that, I decided to come closer to the Mexican border, a little bit of homesickness, to do my PhD in Sanford Burnham Prebys Medical Discovery Institute, where I studied HIV-induced brain injury. And there's a picture of me in my graduation together with my thesis committee. Currently, I am a postdoctoral fellow at the University of California, San Diego, where I study neurodegenerative diseases. So what are neurodegenerative diseases and dementia? Well, neurodegenerative diseases such as Alzheimer's, Parkinson's, and prion disease are a growing burden for global health. Currently, there's around 55 million people that suffer from dementia worldwide, and it is the 7th leading cause of death. Now, it is very important that we understand what is happening with neurodegenerative diseases, because there is currently no cure or therapeutics to slow disease progression. There has been a lot of news lately about a possible therapeutic, but it would be the first of its class to slow down disease progression. However, there's still a lot to be known, because as our population ages, we will have more people develop these diseases. It is expected by 2050, neurodegenerative diseases will be either the first or second cause of death. So it is an urgent matter to study to better design therapeutics. But what are neurodegenerative diseases? Well, is when a healthy brain, either because of aging or some other processes, becomes diseased, starts having damage. And clinically, how does this look? Well, early on, there is changes in mood and behavior in the patients, followed by memory and a cognitive decline. And depends on where the damage is occurring in the brain. There will be some visual disturbances or motor symptoms. And at very late stages of the disease, there will be full-blown dementia. Some of the risk factors associated with neurodegeneration include aging, as well as lifestyle choices. So how much you exercise, what you eat, how much alcohol and coffee you consume can contribute down the road to neurodegeneration or the onset of neurodegeneration. Another factor that can have a big role in neurodegeneration is, for example, genetic factors. So there are types of neurodegeneration that are inherited from one family to the next, from parents to kids. And actually, during the SARS-CoV-2 pandemic, as well as during the HIV epidemic, we realized that viral infections can also lead to neurodegeneration and to damages to the brain. However, what is causing neurodegeneration? That's where I am investigating. So one of the common factors in most neurodegenerative diseases is protein aggregation. So if you think of the brain, of the healthy brain as a machine, these are all the different pieces that are working together in synchrony. However, either with age or mutation or risk factors, proteins can become misfolded. That means that the protein goes from its normal shape to an aberrant shape, and that can start causing problems in the brain. These proteins also have the ability to convert proteins that are the same and make them also take this misfolded form, and they start aggregating together. And eventually, this can lead to damage in the brain. Now, this is a great analogy, but now how does this look in a more disease-relevant context? Well, you have the healthy brain, which after that, you start having this misfolding, we start forming monomers. These are the individual proteins that are misfolded. Then they start aggregating into oligomers, so they start forming like these clumps together, and then they can structure into fibrils. And there is evidence that all of these three types of protein aggregates can contribute to disease progression. Some famous type of these proteins that go misaggregation include tau and amyloid beta in Alzheimer's disease, as well as alpha-synuclein in Parkinson's disease. And in prion disease, there is PRPS. Now, what you're seeing here on the screen is the brain of a patient that was stained for all of these different proteins, and we can see how the different proteins start aggregating and forming these clumps that they shouldn't be forming. However, how do these aggregates impact neurons? So normally in a healthy brain, what neurons do is they first transmit information in an electrical matter through their axons. And then when they come to this junction between two neurons, this is called a synapse. Now in this, the neurons communicate with each other using chemical signaling. However, when you start having these oligomers, these protein aggregates, you start having a lot of problems in the neurons. When we go to the very extreme of when the brain is fully diseased and there is damage, what we have is that there is loss of synapse. So then a lot of these connections between the neurons are lost. There is also a lot of inflammation in the brain. And in the very terminal stage of the disease, you can have full-blown neuronal death. So the neurons die. Now, it is also important to understand that a healthy brain doesn't go straight into dementia and disease. There are intermediary forms. So whenever they will start having accumulation of oligomers, we start accumulating damage over time. How does this look clinically? Well, early on, it has been shown that patients can develop mood and behavior changes, followed by memory and cognitive decline before they reach full-blown dementia and terminal disease. And it is these early stages on disease that I am interested in studying, because we think that this is the area when we can develop therapeutic interventions to prevent patients from reaching dementia or to slow down that disease progression. And the reason why we're interested is, well, early in these intermediary steps, you start having accumulation of proteins, oligomers, and you start seeing synapse loss. Now it's interesting because synapse loss is one of the earliest changes that we see, and synapse loss correlates with onset of disease and clinical signs. And this is before the neurons die. And that is important because it is really hard to replace a dead neuron and impossible to bring a dead neuron back to life. But we think that maybe we can develop, if we understand the disease, we can develop therapeutics that can restore some of the neurons that are damaged back to healthy state, or at least slow down their breakdown. So what I want to study, and the question that I ask in my research, is what are the mechanisms driving early synaptic loss in neurodegeneration? Now, in order to answer these questions, I look at prion diseases. So what are prion diseases? Well, prion diseases are rapidly progressive, incurable, and transmissible neurodegenerative diseases. And these are caused by the protein, the cellular prion protein that I will be calling PRPC. So prion diseases are with the PRPC, which is the normal protein, becomes misaggregated and starts forming oligomers as well as fibrils called PRPS. Now, there has been several outbreaks of prion disease in humans, as well as cattle and cervix. And for human prion diseases, there's three types of human prion disease. First is sporadic. These are diseases that occur without any known risk factor. So these are the most common one, and we still don't fully understand why they're happening or how why the prion protein starts misaggregating. We also have genetic types of prion diseases that is inherited from parents to kids, and this is caused by a mutation on the gene that produces the cellular prion protein, and the gene is called PRMP. However, prion disease is very interesting because it can also be acquired. So if a person can be infected by exposure to abnormal prions, and there's cases of this that happen when surgical tools were not properly cleaned or were contaminated with prion diseases. Or, for example, what is very one of the most known outbreaks is mad cow disease. So when people ate meat from cows that were infected and had prion disease, they actually develop prion diseases themselves. Now here I have to point out that acquired prion diseases are incredibly rare. They are under 1% of the total prion diseases. So there is no real risk for people getting a prion disease via the acquired method anymore. It's incredibly rare. However, since I'm talking to the University of Wisconsin, I thought I would bring an interesting case in which this characteristic of prion disease to be acquired is very important, because you may hear it on the news in Wisconsin, and that is chronic wasting disease. So as I mentioned, cervids, so deer, elk, can develop prion diseases. And right now, the Department of Agriculture, as well as the CDC, are paying a lot of attention to this disease because it's spreading rapidly throughout the United States. And this disease is a prion disease and is characterized by, first, the animals die, unfortunately. They have severe weight loss and the lack of energy. Now, the reason why the CDC is interested on this one is because there is the fear that eventually this prion disease could mutate and jump into humans, particularly if you consume venison or some other type of meat. Again, this is incredibly rare, but it's something that scientists are keeping an eye out for. And it really highlights the need to understand prion disease and know what is happening and how it's leading to neurodegeneration. But jumping back into human prion diseases, how does this look? Well, right now I'm going to show you what a normal brain should look like. And this is an electron microscope. So we use a microscope to look really a high magnification and look at a single synapse. And what you see here, if you follow my mouse, is you see that the synapse is actually slight curvature with a little bit flat. However, when we have the PRPSC, these oligomers, and we infect our models, we actually, in terminal disease, we have neuronal death and synaptic loss. However, the synapses that have not been lost are incredibly curved. So you can see the difference in this other image on an animal that had prion disease that is very curved. And this can be indicative of a lot of neuronal activity. So that means that the neurons are firing like crazy, and that can also lead to damage with too much activity. Now with this, we like to take advantage of prion disease. So we're using prion disease as a model to understand the early alterations in protein aggregation-driven neurodegenerative diseases. So what we do is we take the oligomers either from humans, or not as common anymore from humans, but from other animals, and we inject it into the brain of mice. Now, this allows to see how the disease progresses, because when we inoculate, we call this 0%, and then we go all the way when the animal develops severe symptoms and weight loss, which is we call terminal disease. And at this point, there is severe damage to the brain, and we have this, as I mentioned, 100%. Now, taking advantage of this predictable nature of the disease, we're able to track disease progression. So what's interesting on taking a look at what is happening in these intermediary steps. So we look at every 30 days in disease progression to 150 days. And we call this 20%, 40%, 60, and 80% in disease progression, in which I wanted to understand what are the earliest changes that I can detect. So what I did, if I took the brains of all of these animals, what I'm looking at the screen right now in here is the hippocampus, which is a brain region in the brain. And we stain for the prion oligomers, so the PRPSC in here. And what we see is that already at 40%, so early in disease progression, we start seeing some aggregation, some clusters. Additionally, we wanted to look at ubiquitin. So ubiquitin is a marker that your body uses to tag proteins that are not useful anymore, or that they are misfolded and they need to be destroyed. Now, what we're seeing at 40%, what I observed was that there is actually an aggregation of ubiquitin, suggesting that the body is having problems breaking down some of this junk that is in there and is starting to accumulate it. When we wanted to take a look at the overall structure in the brain, which is staining that we call H&E, we see that the brain starts to develop holes or vacuolations that are characteristic of prion disease all the way until 80% in disease progression. So this is much later compared to some of the changes that we're seeing. Additionally, we wanted to take a look at astrocytes and microglia, which are other cell types in the brain, and that they're in charge of inflammation and of immune response. And what we see is that they don't become active until 60% in disease progression. Now, what this tells us is that something is happening at 40% in disease progression way earlier on that is independent from inflammation, which was one of the leading hypotheses of what can be causing the neuronal damage, and is earlier from vacuolation. So what we decided to do in this case was, again, go back to our prion model. We inoculated mice, and we stopped at 40% in disease progression. And then we collected the hippocampus of the mice, because this is an area that is involved with memory and recognition, which is affected. And we then wanted to take a look at what is changing during this time. And we decided to look at RNA. So all of the cells, of course, have RNA, which is the instructions that tell your body how to make proteins. So we isolated all of the cells and all of the RNA in the brain, and then we send it for sequencing. This allows to see what RNAs are in the sample that we have, and allows to measure what are some of the difference when we compare to our mock, which is just animals that we're injecting with normal brains, so no toxic factor, and then with the prion oligomers. And what we were able to see early on is that with the RNA-seq, we have this group of genes significantly altered. These transcripts are associated with neuronal activity, are an increase at this early time point in prion disease. Well, why is this important? Well, and what is this telling us? First of all, this is telling us that neurons are, or maybe sending a lot more signaling between them than they should have. And this can contribute to damage. What my current research is, this damage can actually be causing damage to the synapse or making them uncomfortable, and eventually can lead to degeneration and loss of the synapses. What my current research is I'm using stem cells and differentiating it into neurons to better understand what is happening at the human level instead of using the mouse model. And with this iPSC-derived neurons, I can also take a specific look at some of the components of the synapse. So in here we have an image of the synapse in which I am looking at some of the different receptors and some of the chemical signaling that neurons are using to communicate with each other, and we want to better understand this. Why is this important? Well, because with this model, we're trying to understand what is driving synapse hyperactivation and loss, and also, if we better understand what is happening in this intermediary stage, we may be able to figure out how to detect early changes in prion disease. So what my work is doing is, we think that if we better characterize early prion disease, we were able to possibly discover potential biomarkers for detection of neurodegeneration early on, before you may even develop clinical signs. Additionally, understanding prion disease is important because other neurodegenerative diseases behave a little bit like prion disease, and because they form aggregates, but also it was recently discovered that in Alzheimer's, you can take either tau or amyloid, which are other proteins that are known to or suspected to cause Alzheimer's. You can take these proteins and inject them into mice models, and these mice will develop Alzheimer's disease as well. So they behave kind of like prion. So better understanding prion disease will allow us to understand all neurodegenerative diseases, or that's the hope that I have with my research. And hopefully when I open my own lab, I can study some of this stuff. And with that, I would like to thank, of course, Daniel and Claudia for inviting me today, as well as the Sigurdsson Lab, which is my mentor at UCSD, all of my collaborators at UC San Diego, as well as Case Western and UCSF for providing us with some of the samples for my research, as well as my funding agency of the UC President Postdoctoral Fellowship Program. But now a fun fact a little bit about myself. So if you guys have ever heard about the movie Coco, it was actually inspired by my home state of Oaxaca and their tradition of the Day of the Dead. So if you ever are interested, and that is Oaxaca on the map, if you're ever interested on beautiful colonial architecture, fantastic traditions, and the carnival that we have in July, as well as just a sweeping view of pyramids at the top of a mountain, I would invite you to visit Oaxaca. It is a great place to be, and I look forward to be able to visit again soon. So with that, I would like to thank you all for coming today to the talk, and I will take any questions that you may have.